In order to perform a LAESI mass spectrometry imaging experiment on a biological tissue or biofilm, the LAESI setup is prepared for robust operation. The next step is mounting the sample onto a sample stage and identifying experimental conditions required for the sample under investigation. Then the lateral LAESI molecular imaging experiment is started and the required data are used to obtain spatial molecular distributions. The results show the molecular architecture of biological tissues and biofilms.
I'm Dr. Peter Nemes from the Vertis Research Laboratory at the George Washington University. The main advantage of the LAESI technique over other methods, including confocal optical and mass spectrometric imaging methods such as MALDI and SIMS, is that the sample analysis can be performed directly on the tissue under atmospheric pressure conditions and no sample treatment is necessary.
Mount tissue sections of interest directly onto a flat surface, such as a chemically pre-cleaned glass slide without chemical modifiers. Secure the sample to a Peltier cooling stage immediately upon mounting. If needed, use a heat sink equipped with a low-power fan to facilitate heat removal from the Peltier stage. Then, if working in a humid environment over an extended period of time for about 1 to 2 hours, inspect for condensation of water or ice on the tissue surface. Water condensation on the tissue detrimentally affects the imaging performance in LAESI experiments. If needed, use a room dehumidifier or place the cooled sample in an environmental chamber filled with an inert gas, such as dry nitrogen gas, to prevent condensation. After optimizing the conditions for maintaining the sample, work to optimize the ion source used. The laser ablation electrospray ionization, or LAESI ion source, consists of a mid-infrared laser, abbreviated mid-IR, and a series of optical elements for light steering and focusing. The setup also includes additional sample holders, cooling components, translation stages, and an electrospray system. A portion of the particulate matter expelled during mid-IR ablation coalesces with the electrospray to yield charged droplets seeded with molecules and ions of the sample. The ions released from these droplets are analyzed and recorded by the mass spectrometer, the orifice of which is shown here. The LAESI setup is configured before starting the experiment. Precise adjustments of the experimental parameters are required to accommodate the sample of choice. To optimize the ion source, start by positioning the sample 15 to 20 mm below the orifice of the mass spectrometer sampling cone. Operate the mid-IR laser at 2.94 micrometer wavelength and 10 Hz repetition rate. Attenuate the laser output to around 100 microjoules per pulse energy. Use a combination of gold mirrors and a focusing lens transparent at the laser wavelength to couple the laser light energy into the sample at normal incidence. Position the mid-infrared beam axis 5 to 8 mm in front of the orifice of the mass spectrometer sampling cone. Adjust the focusing lens position and the laser pulse energy to achieve tissue removal in the focal spot to the desired depth. The dimensions of the ablated volume determine the pixel size for the imaging application. Position a nanospray emitter in line with the inlet axis of the mass spectrometer and at an orifice to emit a tip distance of around 10 mm. For the electrospray, prepare a 50% methanol solution with 0.1% acetic acid for positive ion mode or 0.1% ammonium acetate for negative ion mode. The stability of the electrospray is crucial for successful imaging. Depending on the solvent selection, the flow rate and the spraying voltage need to be adjusted to achieve stable spray. For reactive LAESI in imaging applications, the electrosprayed solution may contain reactants. Use a syringe pump to deliver the electrospray solution through the electrospray emitter at a flow rate of around 300 nanoliters per minute. If the mass spectrometer orifice is kept at a low voltage, for example below around 500 volts measured against ground, generate electrospray by applying high voltage, for example 3000 volts, directly to the electrospray emitter or through a metal union. Operate the electrospray source in cone jet spraying mode for the most efficient ion generation by LAESI. Carefully adjust the relative distances of the LAESI setup to optimize for LAESI ion yield, while keeping the laser beam, the emitter, and the orifice axes in the same plane. With an optical microscope, determine the lateral dimensions of the ablation crater on the sample. For three-dimensional LAESI imaging experiments, perform ablation with individual pulses and determine the depth of a voxel using, for example, the Z-stack mode in optical microscopy. Now that the tissue mounting conditions and the ionization source are optimized, proceed to molecular imaging. In the imaging experiment, the tissue sample is moved in the focal plane of the laser in the X and Y directions with step sizes larger than or equal to the dimensions of the ablation spot. The spatial resolution is limited by the focusing of the incident laser beam. Select an area of interest on the sample surface and obtain the XY coordinates of the corresponding boundaries. Choose a gridding algorithm with which to raster the sample surface with selected dwell time at each pixel over the area to be imaged. Use a 3-axis translation stage and software that is capable of rastering the sample according to the predetermined grid. Then calculate the total time required for imaging. Start the mid-infrared laser source at a repetition rate proper to produce sufficient signal-to-noise ratio for selected ions in the mass spectrum within the dwell time at each pixel to perform a LAESI lateral imaging experiment. Turn on the electrospray source. Make sure that there is enough solution for the full time required for imaging. Now, simultaneously start the acquisition of mass spectra, the mid-IR laser ablation, and the surface scanning, and collect data. When the imaging run has finished, stop the surface scanning, the mid-IR laser, and the data acquisition. Disable the laser source, turn off the high voltage, switch off the syringe pump, and set the mass spectrometer to standby mode. Also turn off the Peltier cooling electronics and close the inert gas flow if used. Finally, plot the ion intensity signal for a selected MZ value against the absolute coordinates of analysis to obtain lateral or 3D molecular images. These are representative images from lateral LAESI imaging of a 100 micrometer thick coronal section of a rat brain. The section was frozen during the experiment and kept in a dry nitrogen gas environment. The anatomical regions of the brain show good correlation with the molecular image obtained for two plasmalogens with MZ278.559. This is a molecular imaging experiment in 3D, using zebra plant leaf tissue interrogated in the ambient environment. Among other primary and secondary metabolites, azacetine with MZ285.076 was detected at higher ion counts in the yellow sectors of the second and third layers from the top, with a homogeneous distribution in the others. This distribution agreed with the pattern of the variegation seen in the optical image.
After its development, the LAESI technique paved the way for researchers in the field of analytical and bioanalytical chemistry to study the molecular composition and spatial organization of tissues and biofilms with appreciable water content. Don't forget that working with a mid-infrared laser and an electrospray source can be extremely hazardous. Always follow standard laser safety protocols and apply proper shielding to all electrical connections during this procedure.